known as prenolipids. Okay, so that gets into this. Let's see, in my phospholipids, isoprenoids is where we're at. And they're called isoprenoids because structurally they have an isoprene substructure. So there'll be two double bonds. So this is isoprene. And isoprene itself is a lipid. It's a natural product. It's actually made in plant leaves and plays a role in the chemistry of air pollution, of basically ozone chemistry in there. So people study it a lot. But isoprenoids as a general class include a bunch of different molecules, basically have this as a substructure. Okay, so what are the different types? So the structural types. Structural types include the terpenes. This is something very near to my heart. This is what I've spent a lot of my career studying, is the terpenes, plant natural products. Okay, and so that includes C5 molecules that have 5 carbons, which is isoprene. But then we can get more complexity. C10, which is compound, which are known as the monoterpenes. And I'll draw a couple of structures here, and I'll show you some pictures later. So an example of a monoterpene might be this. I'll give you this example because let me show you this one here first. That molecule right there is limonene, is the name of that molecule. But what I really wanted to, by drawing it, I kind of wanted to show you is the isoprene substructures, that you have an isoprene substructure here, and then you have an isoprene substructure here. And they're connected in a head-to-tail fashion usually. So they have two different isoprene groups. The terminology is kind of funky. It's like monoterpenes, you think mono, there would be one, but actually no, monoterpenes has 2 isoprene groups. There are similar kind of molecules with 15 carbons, known as the sesquiterpenes. This includes a compound like humulene, which is in hops. Actually, THC, tetrahydrocannabinol, is highly derivatized, but it arises basically from a sesquiterpene skeleton as well. There's C20 carbons, which are known as the diterpenes. This includes molecules like, pine pitch, the pitch in pine trees, that sticky stuff that you have. It actually consists of 2 main fractions. There's a volatile fraction and a non-volatile fraction. The volatile fraction in most of the pine trees are 10 carbon monoterpenes here. And so that's, and people have isolated that and taken advantage of that, and that's what turpentine is. So if you get turpentine, gum spirits of turpentine, that's a volatile fraction of pine pitch, basically, and it's composed of monoterpenes. What's left over is the rosin, and those are C20 diterpene resin acids. And where you see that is, rosin bag, like a baseball pitcher uses a rosin bag, or a violin player puts rosin on their bow. That's the diterpene resin acids. All right, so these terpenes are typically natural products. And natural products are compounds made by organisms, like unique compounds made by organisms, usually for defensive purposes or something like that. And then a lot of people have basically taken advantage of those compounds and for various applications. All right. The next structural class we'll talk about is carotenoids. And these are C40 compounds. And I'm not going to draw the structure. I'm going to show you some structures a little bit. So these are light harvesting pigments in photosynthesis. So we'll see that when we talk about photosynthesis. From a nutritional standpoint, they are vitamin. It's provitamin A is a carotenoid. And ultimately, that's the source of retinoic acid, which is a pigment used in the eyes to detect light. Next, we'll talk about sterols and steroids. These have 30 carbons, so they're C30, and they contain 4 fused rings, at least derived from molecules that contain 4 fused rings. So that a fused ring system like that makes up what's known as the steroid nucleus. Now I'll go ahead and draw the structure of one of them, cholesterol here. So this is the structure of cholesterol. Now, I mentioned that, all these are isoprenoids, and then they have the isoprene as a substructure, except for maybe in here, it's hard to see the isoprene structure. And that's because this nucleus originally was made in a way that you could actually see this structure pretty apparently, but then there's multiple, tens of different chemical reactions to modify the nucleus, and so you kind of lose some of that. It's hard to go back and map where those 5 carbon substructures are all through this molecule, but you can go back and do that. Okay, so some of the roles of sterols and steroids. In the case of cholesterol, it's is membrane structure. There's also a lot of signaling molecules that are steroids. That includes the sex hormones, adrenal hormones, cortisol and that sort of thing, vitamin D, then the other one is intestinal detergents, the bile acids. So actually what happens is cholesterol ends up being metabolized and oxidized to make a carboxylic acid, and it serves as a detergent. All right, let me slide this over and walk through a few slides that I'll illustrate these molecules in maybe a little bit more detail. And I'll also have a couple stories related to it. Okay, so again, the terpenes, that subclass of molecules, the terpene. Here's the isoprene, the sub thing. Here is another example of a monoterpene, alpha pinene, and in this case, it's like 1, 2, 3, 4, 5 is one substructure, and there's another. Again, well, you start out with this head to tail linking of these isoprene groups. They often will fold up in each other and become modified and have structural rearrangement. So sometimes it's difficult to kind of dissect that original isoprenoid structure out of there. So this is alpha pinene. This is a real common compound in the turpentine of pine resin. Here is tetrahydrocannabinol here. So this is the THC. It actually, there's a carboxylic acid that's actually made before this. So this is a sesquiterpene in here. There's a carboxylic acid. And in order to make the tetrahydrocannabinol, there's a non-enzymatic decarboxylation that happens, and it happens with heat, right? So that's why you basically have to smoke your pot or cook it in brownies or whatever, because then you heat up this precursor, it undergoes this spontaneous decarboxylation reaction to end up with the tetrahydrocannabinol. Humulene down here, that's a compound that's in hops, that's a sesquiterpene. Oh, I mentioned this one. This is Taxol. So this is a drug treatment in a couple of different forms of cancers. And just to kind of show you how derivatized these molecules kind of be. Within here, there is actually a diterpene skeleton. So there is a 20-carbon skeleton that's a diterpene. This compound is naturally made in the Pacific yew tree. And anyway, so you have this diterpene, but then you can see it's modified in a lot of ways. There's a lot of other modifications to that. Carotenoids. So these are the 40-carbon compounds. So here is beta carotene, provitamin A. Here you have this big conjugated system. Here you can kind of see that isoprene substructure a little bit together. And actually, the C40 is made up of two 20-carbon units that actually come together in a head-to-head fashion. So like you've got the isoprene here, you have the isoprene there, and then you've got an isoprene here and isoprene here, and there are also isoprenes around there. So again, the natural role of that is to serve as a light harvesting pigment in the light harvesting complex of photosynthesis, but from a nutritional standpoint, this is provitamin A, because then the body gets converted to retinol, vitamin A. So here's the structure of cholesterol. I just Just put it there again. So the role of cholesterol, we're going to talk about membrane, is basically it's membrane structure. It provides membrane stiffness at lower temperatures. So or at higher temperatures, I should say. So we're going to be talking about membranes, but different membranes have different sort of fluidity characteristics, right? And it's going to depend upon what the composition of the fatty acid chains within those phospholipids. If they're mostly saturated fatty acids, you're basically going to have a stiffer membrane, or if they're mostly unsaturated fatty acids, you're going to have a more flexible membrane. If you insert cholesterol into the membrane, you basically, the other thing that it's going to affect the fluidity of the membrane is temperature. The higher the temperature, the more fluid. The lower the temperature, the less fluid. If you insert cholesterol in the membrane, you basically get a stiffer membrane at higher temperatures. So here's an example. Here's our phospholipid bilayer here, and you see cholesterol inserted into the membrane. The hydroxyl group of the cholesterol sticks down and kind of interacts with more of the polar head groups, and then that big hydrophobic rings, the steroid nucleus is sticking up within the center of that phospholipid bilayer. Okay, so I was going to talk about vitamin D a little bit. So vitamin D is actually derived from cholesterol here. So we've got so there's a few reactions, biological reactions, that aren't catalyzed by enzyme. I made a big point about all these enzymes, all these reactions are catalyzed by enzyme, but there are just a few that aren't. One of them is in the biosynthesis of vitamin D from cholesterol. There's a step that requires UV light for the biosynthesis of cholesterol. Vitamin D is an important signaling molecule for regulation, calcium absorption, and bone development. Okay, so this actually allows me to tell a little story that relates biochemistry and genetics to the evolution of human skin pigmentation. Okay, so to tell the story of this, we're going to look at, first of all, go back and see what the current consensus is, or I don't know if there's a consensus, but at least maybe the predominant ideas about when modern humans evolved. And it's about 200,000 years ago. Some people say it was earlier than that, but roughly something in there. And they were thought to have emerged in Central Africa, in equatorial Africa, and they were thought to be hairless, okay, and dark skin, right? And the hairlessness and dark skin are probably related, okay? And that the reason is, so the story that I'm going to basically tell you about this in terms of the evolution of human skin pigmentation is the intersection of three different molecules. Melanin, the molecule that makes skin dark. Tetrahydrofolate, we talked about that as an enzyme cofactor, okay? And vitamin D, this is the one that we're just talking about here. And first of all, let's get into melanin. Okay, so what is melanin? It's that molecule there. It accumulates in a particular cell type within the cell known as the melanocyte, and you get it basically you form the pigment. The pigment will aggregate into little aggregates, and that ends up what's resulting in skin pigmentation. All right. So why do humans need skin pigmentation, particularly hairless humans? And it relates back to the folate discovery. Remember I told you the story about the folates? This is this vitamin that was discovered by Lucy Wills here, who had gone to India to study anemia in female textile workers in India, and basically found that they weren't getting a good enough diet, and they were deficient in this folate molecule in there. Well, the thing about folate is, so here are some of the pathways requiring folate. Nucleotide biosynthesis, amino acid biosynthesis, so that's important. Now, how does this relate to melanin production? folate is degraded by ultraviolet light. So if you have folate here and you expose it to ultraviolet light, it'll break down. Thus, if you're now a hairless primate, right, in equatorial Africa where there's high solar flux, and you have, and in addition, you have a lot of blood flow to the surface of skin to aid in cooling. The problem with all that blood going to the surface of the skin is it can be exposed to the UV light, and then it can break down the folates. So the idea is that melanin is an adaptation to protect against the degradation of folates in these hairless primates in an area of high solar flux in northern Africa. So the original humans were thought to have melanin, be hairless, and then have melanin. And you can imagine, actually, because folates are so important in terms of, particularly for pregnant women, right, where they have a lot of during pregnancy, that there would be strong selection for anything that would help maintain folate levels, basically, so such as melanin levels. Okay, so that's one sort of explains maybe why the original human beings have melanin. Okay, then the next question is vitamin D. So vitamin D is involved in calcium and phosphate absorption. If you don't get enough vitamin D, you end up getting rickets. Okay, and there's a certain symptom, basically, morphological symptom of poor bone development. Most of the vitamin D we actually get these days, it's another sort of public health measure. It's supplemented in the milk. So if you look at raw milk, you won't actually see any vitamin D. But if you get your fortified whole milk in here, it has some vitamin D in there that's been put in there as a public health measure to make sure everybody gets enough vitamin D and we don't have rickets. Okay. The other aspect relating to skin pigmentation is that, just as I mentioned, vitamin D actually requires UV light. So now we've got this folates are degraded by UV light, but vitamin D requires light. But sort of the idea is that if you have human populations in equatorial regions with high solar flux, the potential damage of UV light to folate outweighs the need. You still basically get enough UV light in order to be able to get UV light in order to have vitamin D biosynthesis. But if you didn't have the melanin, you'd have too much and it would break down the folates. But then we get, maybe 70, 50, 80,000 years ago, human populations began migrating out of Africa here, someplace in there. And this is a wonderful story. I don't know if you've ever gotten in this or taken an anthropology class. It's pretty cool. It's pretty interesting. Modern human beings, Homo sapiens, began to migrate out there. They started displacing the Neanderthal populations that were in Europe the whole time. And they also probably displaced the Denisovans that were out in this area. And over, basically from 80 to 20,000 years ago or so, managed to populate the entire planet. There's a big debate over exactly when they came to North America. I'd say the consensus is probably around 20,000 years ago, something like on that. But in doing, though, they moved out of this equatorial region up into these areas of high latitudes. And when the areas of high latitudes, this also represents sort of when the last ice age was receding away from here, so that opened up more space basically in the northern latitudes. And as human beings began to expand in here, they moved to areas that didn't have such high solar flux, right? And so now, when you don't have areas that you don't have high solar flux, you no longer have to worry about the UV degradation of folates, because the sun, the intensity of the sun, and it's the other thing around is you do have to worry about getting enough UV light to be able to make vitamin D. So the idea is that as human populations moved into these high latitude areas with less solar flux, they began to have mutations in which pigmentation was lost. And the interesting thing is, if you follow the pattern of human migrations, this is an adaptation that happens several times. And so there are different genes ends up muting to cause lack of melanin production in different areas. So in Europe, these are the names of some of the genes that were actually mutated that resulted in loss of melanin production. In East Asia, these are different genes. So, I mentioned like me and my kids are Asian, they're adopted, they're Asian. Their whiteness is due to different genetic lesions than my whiteness is. Anyway, so that's a kind of a nice story that relates. I like how the biochemistry and the human evolution overlap with that. All right, now we're going to get into lipid transport. We have a problem a little bit. We as human beings or other mammals or many animals, I guess, have a problem in that much of our system is an aqueous environment, right? The cytosol is aqueous, our blood is aqueous environment. But we also have a lot of these lipids, and we have to transport these lipids around. But by definition, almost lipids aren't soluble in the aqueous environment. So we have to figure out some way we can actually transport lipids through the system, through the aqueous system. So this last section focuses on transport of lipids by a class of molecules known as lipoproteins. Now, what the heck are lipoproteins? Let's first look at lipoprotein structure. So the generic lipoprotein has on the outside a phospholipid monolayer. Not a bilayer, but a monolayer. There's a lot more than I'm drawing here. The other thing it has, embedded within there, are proteins. Maybe here's another protein. There's usually a lot more of those phospholipid molecules around there, but just to kind of get the idea, we will have that. So lipoprotein, we have a phospholipid monolayer with embedded proteins. Then on the inside are tags, triacylglycerols, triglycerides, and cholesterol esters. Actually, it's cholesterol esters. So what's a cholesterol ester? So just an abbreviated structure of cholesterol here. You've got that. But the important thing is you have this oxygen down here, and it will be esterified to a fatty acid. So that's a cholesterol ester. So you imagine the cholesterol ester and the triacylglycerols pretty hydrophobic. And so they're in the middle. We've got the phospholipid monolayer, and there's the, the charge and the polar head groups of that is going to basically enable it to be that. And this is in 3 dimensions, and there's proteins sitting on the surface. So that's the generic description of a lipoprotein. And the role of lipoproteins is to transport cholesterol and the triacylglycerols in the lumen there around the different places. But there are different size classes of lipoprotein that play different roles in lipid transport.
I have a question.
Yeah.
So, judging by that arrow, the tags and the cholesterol esters can pass through the bilayer without the proteins, or do they need the proteins of something like that?
So that's a great question, as to what is the mechanism actually assembly of those things, and there are proteins that actually do this, but there's also a lot unknown about that process too.
Can I get a clarification on spelling? Is it E-S-T-E-R-E or C?
How about S?
Okay, there we go. Thanks.
Okay, what are the different lipoproteins? The largest one that I'll talk about are, I'm going to kind of go from largest to smallest, chylomicrons. And they're substantially larger than any of the other ones I talked. And what their role is to transport dietary lipids through the lymph system to other tissues. So they're actually, I should say, from the intestinal cells to the rest of the body. Once all the cholesterol tag is absorbed out of those, then they're known as chylomicron remnants, and they get absorbed by the liver. We won't get into that detail. All right, the next ones. So if I'm eating a nice fatty meal, that fat, the cholesterol fat gets packaged, gets absorbed by the intestinal cells, get packaged in the chylomicrons, and then it gets distributed to the rest of the body. By the way, here's a little trivia. We might talk about this when we get into fat metabolism. The fat in our adipose tissue that we like this, most of that is dietary fat. It's not fat that we've actually made, our liver makes. It's more like when we eat fat, if we don't have any energy need for it, it gets stored away in adipose tissue. The process of the liver making fat and then storing away in the liver tissue doesn't happen very much. I'm not going to say it doesn't happen at all, but it's enough that ecologists, if they want to know what kind of seeds or what kind of plants plant seeds a critter might be eating, they'll analyze the fat composition and they'll find the fat composition of the food source that is similar to that. They can basically use the fat composition as you know, the kinds of fatty acids in the adipose tissue as a marker of the kind of the food that they actually ate. Okay, chylomicrons. So the next ones I'm going to talk about are actually transport through the blood. And the first one I'll talk about are the ones that are made by liver. And so this is VLDL, or very low density lipoprotein. So this transports tags and cholesterol that have been made by the liver from the liver to the rest of the body. So we have the VLDL particles, and then as tags get removed in the rest of the body out of there, then you have a little bit of what's known as an intermediate density lipoprotein, or IDL. Then as more and more tags get removed, and maybe a little bit of cholesterol, but the tags tend to go first, we end up with LDL. So this is low density lipoprotein. And the LDL ends up being very enriched in enriched in cholesterol. So this is transport of dietary lipids. This is the transport of lipids that are made in the liver, and includes, and you basically start out with VLDL, and so as you have more fats in the middle of your lipoprotein, the density goes down. So when it starts out and it's VLDL, there's a lot of the tags, but then as they kind of go down, it gets smaller and the density goes up. The next, the last of the lipoprotein particles I'll talk about is HDL, which is high density lipoprotein. And what it does is it scavenges the cholesterol from the rest of the body and brings it back to the liver.
Does it take it from cell membranes, or does it take it from the diet?
Oh, I think it takes it from the cell membrane. Yeah.
Okay.
This process is known as reverse cholesterol transport. In general, the size, so these are the largest, less dense, and the way I've drawn down here, these are the smallest, most dense. The density becomes important because operationally, when people would isolate these things, they would do it by centrifugation, and the different particles would migrate in a centrifuge state, would migrate depending upon their density. So the most dense ones would migrate the fastest, and the least dense ones would migrate the least. And so that's how they ended up getting classified by density, because operationally, when you're isolating them, you're isolating as based on how they fractionated via their density. All right, let me have a few yeah.
Pertaining with that, like, if most of it's, I mean, since most of it's dietary, how would like kill this? Is it just too much of it, or like pressure clogs, or is it?
Yeah, so the idea is, how does cholesterol kill us, basically? The idea is that, so it's a risk factor for cardiovascular disease. Cardiovascular disease is the blockage of the arteries, basically, by stuff building up. And if you look at that stuff, this plaque, atherosclerotic plaques, in there is a lot of cholesterol. So I think that sort of the idea is the excess cholesterol just begins to accumulate. It's one of the components in these plaques, but it's a major component that begins to accumulate, and then that slowly makes the size of the artery smaller and smaller. You end up with a heart attack. Okay, so here's some other figures, and I'm just going to kind of re-go over, and I'm going to also show you a little bit of the data behind relating different cholesterol fractions to cardiovascular risk. We'll talk about that a little bit. Okay, so here is a lipoprotein here. And again, the generic structure, the phospholipid monolayer, inside the triacylglycerol and the glycerol esters, and you have some proteins that are embedded in the outside as well. Here's just another kind of figure. And these different particles actually will have different sets of protein in there and slightly different lipid compositions as well. Okay, so then, yeah, the core contains the triacylglycerols and the cholesterol esters. The relative sizes of these different ones. So I mentioned the chylomicron is the biggest, and it's substantially bigger than the other ones. Then we have the very low density, the intermediate density, the low density, and the high density. If you take your blood and you isolate, these are the ones that would be in your blood. The chylomicrons going to be in the lymph system, and you isolate the most abundant ones are going to be your HDL and your LDL particles. Those are going to be the most abundant particles in your blood. Actually, LDL is going to be the most abundant in here. I don't like this. It's just details on the density and that sort of stuff of these different particles. Okay. So we talked about the role of the lipoproteins in the chylomicrons deal with dietary lipids. The VDL deals with lipids that are synthesized by the liver and involves, and then it ends up getting converted to IDL, then LDL as the triacylglycerol is removed. And then the HDL is involved in reverse cholesterol transport. I won't go through this. This basically is a complicated way of saying what I just said. Oh, so here was the question of how. How do these things actually? This is the chylomicron assembly. So this is basically here. We've got absorption of the lipids. This is basically the assembly of the chylomicron. All right. So, cholesterol, as you probably is particularly LDL, is correlated with cardiovascular disease. And so, what's the story about this? First of all, I always like to show this as, why is there so much focus on heart disease? There's a focus on it because it's one of the main things that kills people. If we look at top 5 causes of death by age, 2017. This is kind of fun to go look for. Like, okay, most of you guys are someplace in here, some like this. Unintentional injury. I'll show you some data about what that really means. Suicide, that's kind of sad. Homicide, that's kind of sad. In there. Get here, I'm in the cancer range right here. Cancer is gonna get here. But then in a couple years, I'm gonna enter the heart disease phase here, and we're gonna get in there. But overall, the leading cause of death is heart disease, then cancer, the unintentional injury. I thought I'd go ahead. This is peripheral, but I thought I'd go ahead and talk a little bit about unintentional injury, since that's the one that you guys seem to be most associated with. So these are injury deaths, causes of injury death down here. Car wrecks, kind of understand that. Car wrecks. Unintentional poisoning, that's drug overdose there, in there. Then there's the homicides, firearm, and that sort of stuff in there. I'm headed towards unintentional fall. I think about that when I'm putting the Christmas lights on the roof of the house. Okay. So anyway, get back to heart disease. So question is, what are the risk factors for heart disease? So the way this was really examined was through these large cohort studies, and probably the most famous and earliest one is known as the Framingham Heart Study. And it's named that because it took place in Framingham, Massachusetts. Framingham is a suburb of Boston. If you run the Boston Marathon from Hopkinton to downtown Boston, you run right through Framingham, Massachusetts. And what they did is they basically got a whole bunch of people to sign up, and they did a bunch of blood tests on them, health surveys, and all that sort of, and they followed them for decades. And they basically see, okay, what did people die from? And then what are the risk factors associated with that? And from that, they identified some specific risk factors for dying of a heart attack. And what are some of those risk factors? BMI greater than 27. So if you're overweight, if you're smoking, right? If this is the one that's related to this discussion, if your total cholesterol, all the cholesterol was greater than 220 milligrams per deciliter. If you have diabetes, if you have high blood pressure, hypertension, and then if you start combining these, that it compounds upon itself. So what's going on with the cholesterol one? So this is cardiovascular risk is correlated with total serum cholesterol. So this is the total cholesterol, total serum cholesterol here. Okay, and we see, let's see, all-cause mortality. The higher cholesterol goes up, the greater your risk of mortality is. It can be too low as well. Usually when situations are like this, somebody is sick for some other reason that's causing their cholesterol to get really low. Of course, a big one of that is coronary heart disease. Okay, but here's the thing, is if you look at that distribution, you say, okay, well, I got to get my cholesterol down. There's a lot of people that have high cholesterol that never have heart attacks, and there's a lot of people that have low cholesterol that still get heart attacks. So this is a histogram, basically, of cholesterol in people that have coronary heart disease or don't have coronary heart disease. So this curve, this is the histogram for the people with cardiovascular disease, right? And if you look at the average is going to be someplace in here, and it's greater than this histogram of people who don't have cardiovascular disease, right? So yeah, total cholesterol is associated with cardiovascular risk, but there's a lot of people up here that have high cholesterol that don't have cardiovascular disease, and there's a lot of people down here who have cardiovascular disease have low cholesterol. So what's going on there? Well, one of the things that's going on there is that total cholesterol is probably too crude of measure what the real risk factors are. So we talked about these different places where cholesterol resides in the blood, and then most of the cholesterol is serum cholesterol is associated with either LDL particles. Actually, most is associated with LDL particles, and then HDL particles. And it turns out that whether or not the cholesterol is associated with LDL particles, this is known as the bad cholesterol, then that's directly associated with cardiovascular risk, increased cardiovascular risk. But cholesterol that's associated with HDL particles actually has the inverse effect. The higher that your HDL cholesterol, that's associated with less cardiovascular risk. So this is how that is. So what we have is, on the y-axis here is overall cardiovascular risk. On this x-axis on here is LDL cholesterol. And we see that as we basically go from the left to the right, with increasing LDL cholesterol, we have higher cardiovascular risk. If we look at HDL here, now we're going, from the back is low and the upper is high. As we get lower and lower with HDL, we have increased cardiovascular risk. So the effects are opposite. So when you have the total cholesterol number, it's confusing the LDL with the HDL. Now it turns out there's more LDL than there is HDL, so the LDL cholesterol is kind of swamping out the HDL effect a little bit. But that's sort of one of the nuances, basically, that total cholesterol really isn't the best measure. So here's another just set of data illustrating the cardiovascular risk relative to HDL levels. So the higher the HDL that you get, the lower your cholesterol. So how do you get high HDL? This is what is always problematic for me. You know how you get higher HDL? Exercise. That's what does it. You wouldn't know it by now, but when I used to be a runner, a big-time runner. I ran the Boston Marathon a few times. Anyway, and my HDL was fine right then. But you stop the running and everything like that, then the HDL falls off. All right. So one of the other aspects of is all this stuff is correlative, right? You know, you don't necessarily know is it that HDL and LDL are really the causative factors to influencing cardiovascular risk, either positively, negative, or are they just markers? So, example, HDL and exercise. You know, maybe it's just HDL is a marker of how much exercise you get, and there's all sorts of other benefits of exercise that have nothing to do with cholesterol. That's really improving your cardiovascular risk. Same thing with LDL. Is it really the LDL cholesterol, or is that just a marker for something else? Well, one of the ways you can do that, try to get at that question, is essentially do an experiment where, well, let's try to change the LDL cholesterol levels and see if our cardiovascular risk goes down. So if we try to decrease our LDL cholesterol, do we actually get decreased cardiovascular risk? And that is an experiment that has basically been going on for the last 40, 50 years with statin treatment. So people like me, I'm on statins, right? And basically you take statins to lower your LDL cholesterol. And then you say, well, okay, that's great, but is it actually lowering cardiovascular risk? So this graph here is taken from a review article that looked at multiple different clinical trials of different statins. And what we actually see is pairs of data. So we'll look at this data right here. So this 4S, that's just sort of a code for a particular clinical trial, 4S trial. I don't know exactly what it was. The P means this is the placebo. So this is the people who weren't getting the statin. Then we find its pair over here, 4SS. These are the people that are actually getting the statins. And so what we saw, the effect of the statin was to both lower LDL cholesterol here. So we lowered LDL cholesterol from there to there, and that resulted in lower cardiovascular risk. And so here's another one I guess pointed out. This is lipid P. So this is the placebo of this other clinical trial, the lipid clinical trial. and then here's the statin. So again, the LDL cholesterol was lowered, and your cardiovascular risk was lowered. And one of the things that's been noted is it's hard to get too low, basically, according to this type of study, that you're still seeing the effect even at relatively low cholesterol levels. So that always gets the question is like, well, how low should you go? How low, how much statin should you take? And that's an current area of discussion. So again, it helps in thinking about cause to think about a mechanistic effect. So this is a question kind of you were getting at. What is cardiovascular disease? It's the buildup of atherosclerosis, of atherosclerotic plaques on the inside of the arteries feeding the heart, and you basically end up blocking the flow of blood to the heart. So here's a picture of an artery that has had a lot of atherosclerosis build up around it. And again, this is pretty enriched in cholesterol. Here's some other pictures. This one, this one's pretty bad shape, right? Not much blood getting through there. Okay, so I'll tell you, so one of the reasons I have this data is because my wife, she studies apolipoprotein A1, which is a protein in an HDL particle. And she studies the structure of it and the effect of different amino acids on the structure of that. So she used to get her apolipoprotein A1 particles by getting expired serum from UC Davis. So UC Davis has their blood bank, but it's only you donate blood, it's only good for some, and if it doesn't get used, it becomes expired. And she would go get the expired blood. Or serum, more specifically. Serum is the blood with the red blood cells removed. So this might be typical serum. And then she'd go through a process of purifying HDL particles from that. But occasionally she would get something like that would look like this. It's like, what the heck is that? That is somebody who donated blood after eating a fatty meal, basically. And she likes to use this picture to illustrate we are what we eat in a lot of ways. That it kind of goes in the mouth and we don't think about it, but there actually are these sorts of things. All right, let's take a break there, and we are going to come back and actually before I do that, just one other little tidbit about causality. So we had pretty good data there, looking at causality of LDL in terms of cardiovascular risk, right? If you were able to, through pharmaceutical treatment, lower your LDL levels, then it actually lowers cardiovascular risk. It's suggesting that the LDL cholesterol is actually causal in terms of cardiovascular disease. The other question is HDL causal in protection from cardiovascular disease? And so people assume that it probably was, and so there's therapies have been developed to actually try to people who are of a particularly high cardiovascular risk, infuse HDL into them. And there are also certain genetic variations of human populations that are particularly less prone to cardiovascular disease, and they have a different sequence of the HDL, of the proteins that are in the HDL particles. And so the idea was, well, maybe if we take sort of like these super healthy HDL particles and then inject it into people, that you could actually lower cardiovascular risk. Those studies have not been, nobody's actually been able to demonstrate a beneficial effect of that to date. And that's kind of reverberated through the community, through the sort of the apolipoprotein and HDL community, because everybody begins their grant proposal with talking about how important HDL is to cardiovascular health, and we need to figure out a way to harness that to improve cardiovascular health. And now here's this data that's saying, well, maybe it's just correlative, maybe it's not actually causative. So anyway, that's another little side story. Okay, let's take a break. Let's meet at 20 till, and we're going to